I'm Jennifer Graves, the director of the UC San Diego Neuroimmunology Research Program. I'm a MS neurologist as well as a neuro-ophthalmologist, and I'm pleased to present over the next hour what I think is one of the most critical topics for having a better understanding and developing better treatments for the progressive forms of MS, and that is the role of biological aging. There are a few slides here about the accreditation for this course. This course has received a 1 credit AMA Category 1 for physicians, as well as 1 contact hour for nursing. And this is the information for accreditation for pharmacists as well as PAs. Psychologists and social worker information. I have no disclosures directly related to the content of this presentation. My unrelated disclosures are available on the course information. Some of this work that I'll be presenting today was supported by a generous grant from the National MS Society. The learning objectives for this hour include being able to recognize the role of chronological and biological aging on MS disease progression, becoming familiar with the utility and risks of using DMT in older patients and when you should consider stopping DMT in the older MS population. Identifying comorbidities that impact disease management in multiple sclerosis. And lastly, we'll describe some features of aging and the impact of aging on remyelination and possible ways to reverse those effects. As you know, multiple sclerosis has a very heterogeneous phenotype in different individuals, and age contributes to that heterogeneity. There's a spectrum of severity. In most adults, about 85% of adults will present with a relapsing form of the disease, the remainder with a primary progressive form. Most of you will be familiar with this plot on the right-hand side depicting a typical trajectory of an average adult going through a disease course with MS. The dark black line in the middle represents clinical disability, and the upward deflections indicate acute relapses followed by a remittance and return to close to baseline. On the far right side, you see the onset of progressive disease where the relapses diminish, but there's insidious continual worsening of disability over time. That represents the secondary progressive phase of the disease, or for those with primary, this is where their disease starts. The dotted line at the top indicates brain volume. On the bottom, the arrows indicate relapses or new T2 lesions. And the red line indicates a cumulative T2 lesion burden. Now, if we look at extremes of age, childhood MS, pediatric onset MS, we don't see progressive forms of the disease. 99% of children present with a relapsing form of multiple sclerosis. None of these patients typically present with primary progressive, and we see very little of secondary progression while those kids are still in childhood. If we look at the other end of the spectrum in adults greater than 45 years, we instead see some predominance of the progressive forms of disease. While many adults over 45 or 50 still present with relapsing disease, they tend to land with this first red arrow on the right, where they have a shorter time period in the relapsing phase and will more quickly progress to secondary progression. Or we see 45 years as the mean age of onset for primary progressive MS, the right arrow, where they don't have preceding acute relapses, but just develop an insidiously progressive form of MS. Relapse rates differ by age and notably diminish as individuals are older. In a sub-analysis of fingolimod trials, in the placebo arms, the relapse rate for 18 to 20-year-olds in the 2 trials were 0.74 and 0.60. For 20 to 30-year-olds, the annualized relapse rate was 0.57, 0.48 in the 2 different fingolimod trials. And the total population in this trial, the majority of which were over 30, 75%, the annualized relapse rate was 0.4. This is work that was published in MSJ by Jutta Gartner in 2018. If we add together data from pediatric MS in this graph on the bottom, what you can see is that children compared to young adults, compared to older adults with MS, have declining annualized relapse rates. If we look at this a different way, this is data published by Helen Tremlett in 2008. 2,500 MS patients with close to 12,000 post-onset relapses were studied. These were captured before 2003, before the higher efficacy drugs were introduced. And what you see is overall for all patients, there's a gradual decrease in relapse rate over time from onset of disease, 17% decrease in relapse rate per 5 years of disease duration. But the slope, the rate of this decline in relapse rate is greater in older patients. So let me orient you to this figure on the bottom. On the y-axis is change in relapse rate. For most time points, a -% change in annualized relapse rate. There are 4 columns that represent ages at onset, less than 20, 20 to 30, 30 to 40, and on the far right, 40 plus years at time of onset. And then the individual tick marks check off different 5-year intervals of disease duration. So for all 4 groups, relapse rate declines with disease duration. And again, this is before the use of some of our higher efficacy relapse rate medications. But for the older group on the far right, you see a fast rate of decline of relapse rate. These prior slides beg the question then, if biological aging is a driver of a switch from a relapsing to a progressive form of multiple sclerosis. This is a photograph of the Fountain of Youth, claimed to be discovered by Ponce de Leon in Florida. And while we may not need a Fountain of Youth to be able to reverse the processes of progressive MS, trying to understand key aspects of biological aging, which could contribute to having a progressive MS phenotype, may help us develop new treatment approaches. Some additional background on aging and MS progression. Older chronological age is associated with a faster time to disability milestones. This has been shown in multiple cohorts and multiple countries throughout the world. Biological aging may contribute to neurodegeneration, and this is quite plausible given a decline in remyelination capacity that's been shown in older individuals and animal models. Altered immunologic responses with age. The process of immunosenescence has been well studied, not only in autoimmune disease, but in infectious disease. We know the immune system changes as people reach the 5th and 6th decades of life. Older age at onset of MS means a shorter time to disability, independent of disease duration. So for example, if you have pediatric MS that starts at the age of 10, that individual may stay in the relapsing phase for 20 to 30 years before they reach a milestone of secondary progression. But if the MS starts at age 40 or later, secondary progressive MS may start to occur within 5 to 10 years of disease onset and diagnosis. The mean age onset of secondary progressive MS and primary progressive MS, that we think of them as distinct subtypes of MS, is both around the age of 45. And this is compared to relapsing MS, which is has a typical onset around 30 for men and women. But birth date isn't everything. There's the concept of biological aging as a separate definition than birth date and chronological age. There are large epidemiologic studies that have been performed, including one in the UK that I'm referring to here, that has shown us that from a large group of 38-year-olds who have a birth date that indicate they're 38, that their biological age may range from 25 to 60. Many factors contribute to biological age and this variability. Genetics, the level of exercise and protective environmental actions that patient may take, toxins like smoking, and other environmental and other forms of biological stress. Biological age is very relevant. From that same study mentioned above, that biological age predicted mortality. Biological age is relevant for chronic diseases like heart disease. And we can define biological aging as both somatic forms of aging and reproductive aging. And I'll show you in the next few slides 2 examples of somatic and reproductive aging biomarkers that we could look at in MS patients. So I'll start with the study that we did examining somatic aging in men and women with MS by looking at leukocyte telomeres. Telomeres may be considered the ultimate biological clock. They contain proteins and nucleotide repeats at the ends of chromosomes that shorten with each cell division. Telomere shortening can be accelerated by oxidative stress and DNA damage, and shortened telomeres have been associated with development of cardiovascular disease, dementia, autoimmune diseases, particularly those that tend to peak again in the 5th and 6th decades of life, including lupus and rheumatoid arthritis. And lastly, in one study prior to ours, it was noted that primary progressive MS patients had shorter telomere lengths than those with relapsing remitting MS. The objective of this study was to assess whether biological aging as measured by leukocyte telomere length is associated with clinical disability and brain volume in MS independent of chronological age and disease duration. I completed this work in collaboration with Jorge Oxenberg and the UCSF EPIC team, as well as Ju Lin, who ran the telomere experiments in the Elizabeth Blackburn Telomere Lab at UCSF. The schematic of our hypothesis is listed here below. DNA damage or oxidative stress may contribute, as well as genetic predisposition to leukocyte telomere length. Leukocyte telomere length then triggers the DNA damage response and drives biological aging processes, including the generation of senescent cells and the downstream effects of these cells that, while they can't replicate, can continue to produce pro-inflammatory cytokines. We hypothesize that these processes downstream of leukocyte telomere length could contribute to MS disability. These are the baseline characteristics of the cohort studied. This cohort has been published on previously. This is the EPIC cohort with up to 10 years of follow-up data. Over 500 patients were studied. The mean age was 43, which was quite appropriate for the question of telomere attrition. Some of their other features are listed here. I'll draw your attention to the range of MS subtypes. Most of these patients at the time they were enrolled were relapsing remitting MS. And then at the bottom of the slide, you'll see that the mean TS ratio, which is an expression of leukocyte telomere length, was 0.97. This is on par with what we would expect with a healthy control population in this age range. So we didn't see immediate evidence that MS patients were suffering more telomere attrition than healthy controls. Rather, we wanted to use telomere length as a biomarker of how aging might affect MS disability. And I'd like to acknowledge in the bottom right corner here, Dr. Kristen Crisco, who performed this analysis. We published this work in the Annals of Neurology in 2019, and the primary outcome for analysis was disability, as determined by the EDSS score. And I show you here the multivariable model results for an association of telomere length with the EDSS. The adjusted models accounted for chronological age or birth date, sex, as well as disease duration, so a conservative model adjusting for those other time variables. And we saw that for every 0.2 shortened leukocyte telomere length, there was a 0.27 higher disability score, EDSS, and was highly statistically significant. We also found statistically significant associations with total brain volume and total white matter volume with clinically relevant effect sizes. We performed a longitudinal analysis on a subset from that larger group of 23 pairs of individuals who had their leukocyte telomere length measured at several time points over time. So a total of 46 patients, a smaller subset. But what was critical to these pairs were that these pairs were matched based on their baseline age, disability, as well as disease duration, but one half of the pair went on to develop secondary progressive MS, and the other half did not. And we looked at changes in disability scores in these matched pairs over time. And we found, again, for our primary outcome, an association with disability. For every 0.2 TS ratio shortening of the leukocyte telomere length, there was a 0.34 higher EDSS score for those individuals, and that was statistically significant. There were other coefficients of interest in this adjusted analysis on the right half of the table, but they did not reach nominal statistical significance, and that was in part due to the small sample size of this group. We are currently working on analyses for a much larger data set of leukocyte telomere length measured over time to replicate these results. In conclusion, for this segment of the presentation, shorter telomere length, a marker of biological aging, was associated with higher disability and lower brain volumes. Telomere attrition explained 15% of the variance of the effect of chronological age on disability. Aging-related processes may contribute to MS progression. Oxidative stress, decline in remyelination capacity, the altered immune functions we know that happen with aging could all contribute, and comorbidities and lifestyle factors may also be contributing. Targeting aging-related processes may be a therapeutic strategy for addressing disability and progressive MS. Now switching gears to talk about reproductive aging with ovarian function as an example. We are also interested in pursuing studies and looking at testosterone and andropause concerns for men in reproductive aging. But for the point of this presentation, we'll focus on this data of ovarian function. And we published this work in Neurology in 2018. The mean age of onset of PPMS and secondary progressive MS are both approximately 45 years of age, very conspicuously during a time of ovarian aging and perimenopause. The last menstrual cycle typically occurs in North American women around the age of 51. But ovarian aging is an up to 10-year period of decline in ovarian function. Most of the biology of interest, most of the fluctuations that could be contributing to a change in disease course are probably happening before that last menstrual cycle. Women catch up to men in disability around the age of 50. So we pose the hypothesis that does ovarian aging contribute to the development of progressive disease? To pursue this hypothesis, we looked for a convenient biomarker that would give us a readout of ovarian function. It's very unpractical to do ovarian ultrasounds on a large number of women, and we identified a marker, anti-Mullerian hormone, that ironically is the hormone that makes a male early on in development, but later on is practically undetectable in men, but begins to be secreted by granulosa cells of follicles in females around the time of puberty, reaches a steady state in young adulthood, and then lowers over the course of an adult woman's lifetime, reflective of her ovarian function. It explains at least 82% of the variability of the age of menopause, and it's become a new surrogate for ovarian reserve in fertility clinics. And very conveniently, it doesn't fluctuate much with the menstrual cycle. So unlike estrogen or androgens or FSH, it doesn't fluctuate, and you don't have to perfectly time your blood draws for the time in the menstrual cycle. Very convenient for leveraging large databases and data banks of samples. On the right-hand graph, you can see that different women in different chronological age groups have different rates of decline of this AMH. So you can separate out biological from chronological age effects in assessing the function of the ovaries. When we use AMH and the study of ovarian aging as a potential driver of phenotype in MS, there could be two things mechanistically that we're evaluating. One is the ovarian function of producing hormones, estrogens and androgens, and the loss of those hormones over time in later stages of MS. But the other is that the ovary is like the canary in the mine for aging processes. It's an early marker of the onset of biological aging, and more specific than chronological age in estimating when those effects are starting to occur. The follicular cells in the ovary have 10 times the mitochondria and are much more sensitive to stress than other cell types. That's why we worry about ovarian function in the follicles when we expose patients to toxins like chemotherapies or other toxins in the environment. And it's also sensitive to telomere length. The objective of this analysis was to determine if ovarian decline as measured by levels of anti-Mullerian hormone is associated with clinical disability or brain atrophy in women with MS. So connecting this slow decline over a 10-year period on the left-hand side in AMH with development of disability in MS on the right. To proceed with this analysis, we formed collaborations with Geraldine Messerlin, a reproductive endocrinologist at Brown on the right on this slide here. She performed our AMH assays and is the go-to biomarker lab for the North American menopause studies. And on the left-hand side, excellent input and advice and collaboration with Marcel Cedars, Chief of Reproductive Endocrinology at UCSF. In women with MS, 412 compared to healthy controls were included in cross-sectional and longitudinal analyses with up to 10 years of clinical and MRI follow-up. We sampled the AMH at 4 time points, baseline, year 3, year 5, and year 8 to 10 using an ELISA, ultrasensitive ELISA on plasma samples. We applied appropriate statistical methods, including logistic, linear, mixed model regression, and we adjusted for the following potential covariates. Multivariable models, we adjusted for age, chronological age, disease duration, smoking, race, ethnicity, vitamin D, the disease-modifying therapy, BMI, birth control, or other hormone drugs like hormone replacement therapy. And the final models were determined by backward selection. Not all of these confounders ended up having a significant impact on the model. These are the results of the baseline clinical analyses. Ovarian reserve, and we used as a unit of measurement for the model, a twofold decrease in the anti-Mullerian hormone level was associated with an increase in the EDSS and worsening on the MSFC. Now, this 0.13 increase in the EDSS may not seem that impressive, but if you compare that in the footnote at the bottom of the slide to what you see typically with just 1 year disease duration, it's about 2.5-fold more than what you see with just an additional year of having multiple sclerosis. And both of these results were highly statistically significant and consistent for a baseline association of AMH level with disability level, even after adjusting for chronological age. These are the results of a baseline MRI analysis, again, looking at a twofold lower AMH level with cortical gray matter, total gray matter, and white matter metrics. Again, conservatively adjusting for birth date, chronological age, disease duration, and body mass index. And we saw a likely clinically relevant effect size for cortical gray matter that reached nominal statistical significance. And again, you can compare in this footnote the minus 2.24 gray matter volume compared to what you would expect for just an additional year of having MS at the bottom of 0.6. So relevant effect sizes and statistical significance. Then it's always important to include in your analyses a longitudinal addition to cross-sectional analysis, adds validity to the observations. We performed a longitudinal study of AMH in relationship to the disability score using a multivariable, random intercept, random slope model. It allowed us to use all observations we had on patients over time and to look at both within and between subject comparisons. For every twofold decrease in anti-Mullerian hormone, there was a 0.079 increase in the EDSS, and it was highly statistically significant, further supporting the hypothesis of a potential link between drops in AMH and increases in disability. For the longitudinal analysis of AMH with gray matter volume, we performed a similar type model with all the same advantages, allowing within and across patient comparisons. We also conservatively adjusted for chronological age, disease duration, and body mass index. And for every twofold decrease in AMH, there was a decrease in the gray matter volume of minus 1.6 millimeters cubed, for cortical gray matter minus 1.4 millimeters cubed. And you can see that they did not quite reach nominal statistical significance, but these results are consistent with what we saw in the disability analysis. So in conclusion, for AMH, lower AMH levels as a marker of ovarian aging are associated with greater disability even after a conservative adjustment for birth date, for chronological age, disease duration, and BMI. Lower AMH levels are associated with lower cortical and total gray matter volumes, but are not associated with T2 lesion volumes. These results support the hypothesis that ovarian decline may be associated with an increase in MS severity. There are small trials ongoing examining estrogen receptor agonists and other estrogen-based potential therapies for perimenopausal women with MS, but estrogen is unlikely to be the only factor contributing to ovarian aging's relationship with progressive disease. Many biological changes that occur during this time period for women. And here I'm going to switch gears a little bit to talk about disease-modifying therapies and aging, and how we should approach the issue of treating patients in the 5th, 6th, 7th decades of life. DMTs largely target relapses, and as I showed in the beginning of this presentation, relapses decline with aging. 17% decline in relapse rate for every 5 years of disease duration. Individuals tend to lose the high peaks of inflammatory responses and events that trigger relapses, and then ultimately also trigger regulatory responses. Instead, older individuals may develop a low-grade inflammation that more rarely triggers effective regulatory response that shuts off that inflammation. And these processes of aging and their aging immune system may contribute to poor responses to therapies, to progressive disease, development of comorbid illnesses, and even side effects with our therapies. The benefit versus risk ratio shifts, and we have very little data to use on patients over 55 to guide our decision making. Most clinical trials in MS have stopped including patients at the age of 55 when they enter studies. Immunosenescence and a relative lymphopenia compared to younger adult patients may contribute to greater infectious side effect risks, including PML risks, as well as more common infections, pneumonias, URIs, and UTIs. The timing of when to stop DMT as a patient transitions to secondary progressive MS is not clear and is not laid out yet, but there are increasing amounts of studies addressing this issue, observational studies, and we also have randomized studies that are underway to look at the consequences of stopping DMT in the 5th, 6th, or 7th decade of life. And these include DISCO MS, STOP ICEP, and others that are listed in clinicaltrials.gov. A critical issue, in my opinion, in studying DMT use in older individuals is we may need to use different outcomes. I've demonstrated in several slides now that relapse rate and other studies also demonstrate decline in UT2 lesions as people age. We need different markers of the types of inflammation we expect older individuals to experience with MS. Smoldering inflammation of aging has been reported by several groups. It matches with what we understand about immunosenescence and other autoimmune diseases and other infectious illnesses, and even in cardiovascular disease, our understanding of what happens in the senescent immune system. And radiographically, as well as pathologically, several groups have reported this concept of smoldering inflammation, chronic lesions, rather than just accumulation of new lesions in older patients. And perhaps by pursuing quantitative measurement of smoldering plaques, chronic active lesion, as is shown here as an example from Absinthe et al. And JAMA Neurology, Danny Reich's group in 2019, may help us develop things to use in the short term to look at DMT efficacy in older individuals. What about stopping DMT? Short of having even more data about what the right thing to do is until we have all the results of all the randomized trials, we can look to some of the observational studies. And I want to acknowledge Dr. Hua from Cleveland Clinic in Las Vegas, who was one of the original symposium faculty that I had invited to the session that was to be done in person. And I wanted to highlight some of the work that she was going to present. Greater than 60 may be a safe time to stop currently available DMTs for many patients. Her group studied 600 participants, greater than 60 years of age, 178 of whom stopped DMT. So 600, so they had a control group of those who didn't stop or hadn't been on DMT, and 178 who were acutely stopping. After they stopped, only one relapse occurred in these 178 individuals. Only 10% restarted DMT after discontinuation, and largely for a perceived prior benefit for some of their symptoms. There were no changes in the time 25-foot walk progression between continuers and those who stopped. But there are exceptions to the rule. We certainly all encounter patients over the age of 50 who are still having gadolinium-enhancing lesions and still having relapses. And we know there have been several drugs that have been specifically studied in this group of active or perhaps relapsing secondary progressive MS, or just relapsing MS patients in older age groups. We need to understand biological age in this context. These patients haven't been interrogated for what's happening at the level of the immune system or the rest of the things that we might be able to measure about their biological age. In another study looking at Olmsted County MS patients, 38% of participants in this record-based study had not been transitioned from a diagnosis of relapsing to secondary progressive MS by the age of 75. And we need to better define this group of individuals who may or may not have a slowdown of their relapsing activity, but also may not be progressing significantly on our typical EDSS exams. What type of MS does that patient have when they fall between relapsing and secondary progressive MS, and what's happening at a biological level in those patients? One size probably doesn't fit all for determining DMT choices based on chronological age alone, and we need more biological aging information to make informed decisions about how to manage these patients. I thought I would put one chicken or the egg slide in this presentation, and this felt like the right spot. One issue that comes up when you study aging processes in MS is most of what I presented to you is based on this hypothesis that normal biological aging, aging in the immune system may be driving some of the changes that we see in MS patients, but could MS itself or the DMTs we use accelerate immunosenescence or accelerate aging processes, compounding that problem? Overall, I would say it's not 100% clear to me that this is occurring. One thing that's very challenging in asking this question is that both disease duration and aging, they're tightly coupled. They're time-based variables that are highly related to each other. So it takes a very careful approach to separate out their effects when you're analyzing data. Repeated clonal expansion events in immune cells could certainly drive senescence processes. And some DMT mechanisms of actions have been evaluated, particularly anything that's lymphocyte depleting. Could that contribute at all to senescence? But these concepts, I think, are not proven out yet in the MS population. The LTL study that we did, and I showed you previously, using leukocyte telomere length as the bioaging marker upstream of senescence pathways, was similar in MS in healthy participants. In other words, we did not see accelerated telomere attrition in MS patients beyond what we see in healthy controls. But we could look at specific cell types and dig down a little deeper to see if there is any evidence in certain cell types, memory T cells, memory B cells, to see if there's any evidence of accelerated senescence in aggressive MS patients. Switching now to talk about comorbidity in MS. Very important when making DMT choices and in caring for our patients. Highlighted here are some of the most common comorbidities that have been reported to affect patients with MS. And something to keep in mind is that this biological aging process that could be driving the progressive forms of MS are also drivers for early onset cardiovascular disease. We know that telomere attrition has been linked to atherosclerosis development, coronary artery disease, stroke, heart failure. So all of these factors are important to keep in mind when we're thinking about aging in our MS population. Disability from having MS may increase sedentary behavior with a subsequent risk for development of comorbid diseases associated with sedentary behavior. Obesity is a known risk factor for MS and for many of the other comorbid illnesses, diabetes and heart disease that patients with MS may suffer from. And comorbid illness may limit the DMT choices you have for that patient. And thus, you may have an aggressive patient that you can't place on an aggressive therapy and therefore not be able to maintain disease control. And I'd like to highlight here the work of Ruth Ann Marie, one of the other faculty that was slotted to give a full talk at our symposium, and just wanted to give a brief highlight of that work here. So this is a publication from Neurology in 2016 in a study of over 23,000 Canadian MS patients and over 110,000 matched controls. MS patients had a higher frequency of all the comorbid illnesses they looked at, except for dyslipidemia, and that was at the diagnosis of MS. So the number one comorbidity was depression, with a frequency of close to 20% in MS patients. The second was hypertension, with a frequency of 15% in MS patients. And hypertension, when you looked at women with MS and healthy age-matched controls, 16% higher odds to see that in the women with MS than the healthy matched controls in that Canadian data set. And for men, 48% higher chance to see hypertension in a man with MS compared to an age-matched healthy control. So very notable to appreciate the amount of burden of these illnesses that our patients with MS have. MS participants also had a 17% higher odds to have diabetes and a 30% higher odds for ischemic heart disease than matched controls. Vascular comorbidity is associated with more rapid disability progression in MS. This is work from Ruth Ann Marie et al., Neurology 2010. This is a Kaplan-Meier survival curve. On the y-axis is the proportion of patients without unilateral walking assistance, so the proportion who don't need a cane yet, versus on the x-axis time from diagnosis. In the black line, no vascular comorbidity, dotted line, any vascular comorbidity. And you can see these curves split rather quickly. Well before even 10 years of disease duration, the patients with a vascular comorbidity start developing the need to use a cane more rapidly than those without this comorbidity. So it's critical to diagnose and to treat comorbidities in MS. As appropriate, you can do screenings in a neurology visit. Most of us are at least checking blood pressure for patients that we're seeing in clinic. Also consider requiring patients to closely follow with the PCP in addition to their MS provider to make sure they're not missing out on routine health screenings. Education for patients on not just their MS, but the need to take care of these other medical problems as well. Wellness training for participants, for patients, diet and exercise training, and to make exercise and these wellness initiatives accessible to patients with ambulatory disability or other significant forms of disability that may preclude them from taking part in some of these wellness activities or getting to their other doctor appointments. And lastly, I'll conclude with a few slides commenting on aging and remyelination. I want to acknowledge that Bjorn Newman was the fourth faculty member that had graciously agreed to fly from London to the CMSC meeting, and he works in the Robin Franklin lab, so I'll highlight some of their work in the next few slides. Aging and remyelination, some basic principles. This is outlined very nicely in a review article in MSJ from 2019. There's a failure to remyelinate, and it might be a contributor to age-related MS pathology and progression. In later disease course MS chronic demyelinating plaques, there are undifferentiated oligo-lineage cells, an abundance of undifferentiated cells rather than functional oligodendrocytes. And this suggests there is a differentiation failure in aged OPCs. Remyelination capacity may decrease from both a biological aging process as well as from the stress from disease duration and repeated events of oxidative stress in the brain environment. MRI studies also suggest reduced remyelination in older patients. One of, I think, the very well-known experiments from this lab group, from Robin Franklin's group, published in two different articles in 2012 and 2013, this concept of rejuvenating regeneration in an aging central nervous system. And the experiment depicted in the figure below is that of a heterochronic parabiosis. And in this experiment, they connected the circulatory systems or provided young immune cells such that an older animal was able to receive something rejuvenating from the circulatory system or the cells from a young animal to re-stimulate the ability to remyelinate. They have followed this work with very exciting work looking at the effects of fasting and metformin on the capacity to remyelinate. Age-related failure to produce oligodendrocytes from OPCs can be reversed by fasting as well as by a fasting mimetic, metformin. And aged OPCs treated with metformin regain the ability to respond to differentiation signals. And an example of the remyelination they were able to achieve by applying metformin to aged OPCs is depicted in this figure below. And lastly, I thought this figure that they published in their review article from MSJ in 2019 captured the work quite well. So if you'll start with me in the upper left-hand corner, a young or aged OPC, when exposed to a young microenvironment with more stem cell potential and less aging hallmarks of the tissue, brain stiffness, other factors. If it's in a young environment here on the far left side, then the young or the rejuvenated OPC is capable of responding to differentiation signals and a positive feedback of self-renewal, creation of functional oligodendrocytes and more efficient remyelination. And that's in comparison to on the right side of this figure where the young or aged OPC is exposed to an aged microenvironment, and whereas the young might be able to pull through to some degree, the aged OPC in an aged microenvironment cannot respond to differentiation signals, does not form functional oligodendrocytes, and leads to poor remyelination. And then in the middle of this graph, the heterochronic parabiosis, fasting or metformin can shift that aged microenvironment to a younger microenvironment and rescue or rejuvenate those OPCs for better and efficient remyelination. But what can we do now about biological aging, short of finding a true fountain of youth? Well, exercise is the #1 word here. The one thing Elizabeth Blackburn has been able to repeatedly show that can safely lengthen or maintain telomere length is exercise, healthy diet, lifestyle, avoiding the comorbidities as much as possible, using appropriate screening and treatment for hypertension, diabetes, and heart disease. And we need to continue to study the mechanisms of biological aging in MS, including clinical trials testing these concepts in adults greater than 55, using appropriate outcomes for the types of inflammation and disability progression we expect to see in this population. We may not need this fountain of youth, nor a science fiction human parabiosis experiment, but we need to identify the key players in aging that change MS phenotype. In summary, the host's chronological and biological age matters. It's important to understand how aging processes may contribute to MS phenotype. Studying changes in MS throughout the life cycle may allow us to finally get closer to personalized medicine, to specific treatment strategies and new targets. Shortening telomeres may portend the risk of progressive disease. In adult women, ovarian aging is associated with increased disability and gray matter atrophy. Comorbidities may be both a cause and effect of progressive disease and they are related to biological aging as well. The DMT risk balance shifts as patients get older, and we have to take this into consideration when picking medications. It may be appropriate to stop DMT around the age of 60, but results from randomized trials will further support this concept and let us know what the consequences are for patients when we stop medication. And we need to study adults over 55 in clinical trials more systematically, and we need outcome measures that are appropriate for this group and capture the pathologic processes we think are occurring. Heterochronic parabiosis, fasting, and metformin can change the microenvironment to make old OPCs more receptive to differentiation factors to rejuvenate the remyelination processes. Not everything can fit in a 1-hour CME course on biological aging. There is much more we need to understand and synthesize about the aging processes in neurons and glial cells, in mitochondrial function, in aging, in neurons, and the interplay of all of these processes with the vascular dysfunction that can happen with comorbid illnesses and with immunosenescence. I feel there's a call to action to understand these aging processes in patients and to try to fill in the knowledge and treatment gaps and lead us towards better therapy options for the progressive forms of MS. Many people to acknowledge in this presentation, my current team here at UC San Diego and the support I'm being given in my new home in Southern California. All of my colleagues and wonderful collaborators at UCSF who made the AMH and telomere work possible. The Blackburn Lab, specifically Ju Lin, who ran the telomere experiments. Brown University, Geraldine Masurlian, who was responsible for the AMH assays critical to that work. And also to Ruth Ann, Lee, and Bjorn, who we were very excited to do a full symposium together and look forward to an opportunity to do this again in the future, in person, when we can all gather. And of course, acknowledging the financial support that I had during the time of the work I presented, the National MS Society support, CMSC, and Race to Erase MS. Thank you very much for your attention. This is our research home here in La Jolla, the Clinical Translational Science Unit. If you have questions, concerns, ideas about biological aging and MS, please feel free to reach out. Thank you.